The Krebs cycle is an important part of aerobic respiration that fuels cellular processes. While exercising, the body demands a constant flow of ATP for the muscles under use. Therefore, the primary goal of the Krebs cycle is to harness available energy from acetyl-CoA into the reducing power of NADH and FADH2, which go on to produce ATP in the electron transport chain. The Krebs cycle is also essential for the development of the endothelial system in the fetal period, as an absence can cause heart problems at birth. This occurs in the mitochondrial matrix, the hub of cellular respiration, in an 8-step process. The first step puts energy into the system. Acetyl-CoA is derived from the previous metabolic pathway, glycolysis, by oxidative decarboxylation of pyruvate. The enzyme citrate synthase leaks the oxaloacetate substrate to the acetyl-CoA's acetyl group, releasing citrate and coenzyme A. However, the citrate is too stable for the reactions that follow, so the enzyme aconitase links to the citrate to remove a water molecule, forming a more unstable citrate isomer. Aconitase then reattaches the water molecule, forming isocitrate. With the citrate rearranged, isocitrate dehydrogenase transfers an electron to positively charged NAD, producing NADH and an oxalosuccinate intermediate. Also, a carbon atom is detached to form carbon dioxide, transforming the 6-carbon molecule to a 5-carbon molecule known as alpha-ketoglutarate. This step involves a highly developed complex of 24 enzymes, alpha-ketoglutarate dehydrogenase, which transfers electrons to positively charged NAD, producing NADH. A carbon atom is then detached to form carbon dioxide, transforming the 5-carbon molecule to a 4-carbon molecule. Coenzyme A is then relinked to form succinyl-CoA. Succinyl-CoA synthase links the molecule, releasing coenzyme A and energy, which is used to combine a phosphate to GDP, forming GTP. Succinate molecule is formed. With carbons removed, the rearrangement process occurs and hydrogen is manipulated. Succinate dehydrogenase enzyme links to succinate to release 2 hydrogen atoms, which attach to carrier FAD, forming FADH2. A fumarate molecule with carbon double bond is formed. Fumarase enzyme continues the rearrangement by adding hydrogen and oxygen to the fumarate molecule, forming malate. Finally, malate dehydrogenase enzyme removes 2 hydrogen atoms from malate, which attach to positively charged NAD, forming NADH plus H. The oxaloacetate substrate is then reformed, allowing the cycle to start again. During glycolysis, 2 molecules of acetyl-CoA is formed, enough for 2 cycles. Therefore, the 2 molecules of acetyl-CoA are metabolized into 4 molecules of carbon dioxide, with resultant generation of 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of GTP. The products NADH and FADH2 can produce ATP in a further process of cellular respiration known as the electron transport chain. This requires oxygen and provides the muscle fibre with an enormous amount of ATP. However, in the absence of oxygen, anaerobic respiration occurs and the cycle stops at the Krebs cycle.